Hello students, today we'll continue with the cell biology in the environmental monitoring with part 2. So in the previous slide we were analyzing the use of cell organelles in detection of chemical mutagen, that is carcinogenic or mutagenic agent that are present in the environment. So in the previous slide we had talked about chromosomal damage, use of micronuclear as detecting the damages in the cell, and sister chromatid exchange in the previous slide. So today here we will be exploring about the membrane damage that is caused by the toxins or the mutagenic agent. So one of the important tests that we use to detect is lipid peroxidation. So lipid peroxide is the when there is lipid molecule that are oxidized or broken down into smaller particles. So this will lead to production of super oxides or peroxides in the cell. So these peroxides can damage the protein or can damage the DNA when they are released. So it will lead to oxidative burst in the cellular cell and can lead to cell death and lead to apoptosis. So the measurement of lipid peroxidation is a good measure to analyze the DNA damage or the amount of damage that is caused by chemical mutagen. So here the one of the example or the lipid peroxidation is dictated by the lipid peroxidase enzyme. So that would produce a hydrogen peroxide molecule. So the amount of hydrogen peroxide molecule that are present in the which is eventually broken down into water and oxygen molecule. So the amount of production of hydrogen peroxide is a good measure to detect the membrane damage that is caused by heavy metals. You have organophosphates, then you have various different chemicals, food adulterant, maybe dyes that are used, or food coloring agent that causes these kind of damages into the lipid membrane. So membrane fluidity. So toxic compound can modify membrane structure and fluidity through lipid peroxidation. So the structure is completely damaged and there won't be an a proper cell membrane. So the cell may be shrink. The size of the cell will be much lesser than the original or normal cell. So that is an important criteria for cellular damage. So membrane dysfunction. Dysfunction of membrane lead to cell death and organismic death. So what happens is they release apoptotic protein or apoptotic lysosome is proteins are released, so that leads to death of the cell. So these is the damage that is caused. So there are other ways to detect. So in the previous slides we talked about how cell is used, cell as a whole is used to detect the environmental pollution. So now let us explore the molecular biology in the environmental monitoring. So here the use of DNA probe. So DNA probe is DNA and RNA molecule can be used as DNA probe. So they are complementary sequence that are present to the sequence of the microorganism, and it enables the detection of specific gene. So when there is a damage that is caused by chemical mutagen to a specific gene, we can analyze using DNA probes. Then we have immunoassays. So immunoassays utilize antigen and antibody interaction to detect specific protein, providing valuable information about environmental contaminant. So antigen and antibody interaction is the main component. So many of the chemical mutagen have or elicit antibody reaction when they come in contact with the body or when they are ingested inside our body. So using this specific antibodies or the protein, we can be able to detect the chemical mutagen quickly and efficiently. So, these are some of the methods that are used in detection of chemical mutagen. So, these molecular techniques are widely used in the environmental analysis, including the detection of infectious diseases, identification of pathogen and food contamination. So, by this we can conclude that these molecular method and cellular method are very useful in detecting the chemical pollutants. Thank you.